Today, I'll be presenting on southern blotting, a fundamental molecular biology technique used to detect specific DNA sequences. We'll explore its history, principles, applications, and significance in modern science. What is southern blotting? Southern blotting is a foundational molecular biology technique developed in 1975 by Professor Sir Edwin Southern. Contrary to what its name might suggest, it's not related to geography, but rather named in honor of its inventor. This method paved the way for related techniques like northern blotting for RNA and western blotting for proteins. Southern blotting enables the precise identification of specific DNA sequences within complex samples. It integrates several key steps: restriction enzyme digestion, gel electrophoresis, membrane blotting, and hybridization with a labeled probe. Steps in southern blotting technique. Southern blotting is a multi-step technique used to identify specific DNA sequences. The process begins with DNA isolation, where DNA is extracted from cells or tissues. In the second step, restriction enzyme digestion is carried out. During this step, the DNA is cut into smaller fragments using specific restriction enzymes. Next, gel electrophoresis is performed. In this step, the DNA fragments are separated based on their molecular weight as they move through a gel under an electric field. In the fourth step, the separated DNA fragments are denatured, converting double-stranded DNA into single strands. In step 5, the denatured DNA is transferred from the gel onto a stable membrane, preserving the pattern of separation. Step 6 involves hybridization, where a labeled probe binds specifically to its complementary DNA sequence on the membrane. Finally, in step 7, DNA detection and visualization are performed. This allows the target DNA fragment to be identified and analyzed. In the following slides, each of these steps will be discussed in more detail. The first step begins with the extraction of genomic DNA from the cells. At this stage, it is essential that the DNA is both pure and intact, free from any degradation. To achieve this, all proteins and RNA must be carefully removed, ensuring that only high-quality genomic DNA remains for the processes that follow. The second step involves restriction enzyme digestion. In this phase, specialized enzymes recognize specific DNA sequences and cut the genomic DNA at those precise locations. This process generates defined DNA fragments, breaking the long genome into smaller, more manageable pieces. These controlled cuts ensure that the DNA is prepared in sizes suitable for the analyses and procedures that follow. Now that the DNA has been cut, it's time to separate the fragments. In this step, scientists use a technique called gel electrophoresis. The DNA samples are loaded into a soft gel, and an electrical field is applied across it. Because DNA is negatively charged, the fragments begin to migrate toward the positive end. But not all fragments move at the same speed. Smaller pieces travel faster and farther through the gel, while larger ones lag behind. This creates a distinct pattern of bands, allowing researchers to analyze the size and identity of each fragment with precision. At step 4, the DNA double helix is unwound and converted into single strands. Scientists achieve this by applying an alkali treatment, typically sodium hydroxide, or NaOH. The alkaline environment disrupts the hydrogen bonds holding the two strands together, allowing them to separate. This denaturation step is essential, because only single-stranded DNA can undergo hybridization, where complementary sequences pair up to reveal genetic information. After the DNA fragments have been separated in the gel, the next step is transfer. In this process, the DNA is moved from the gel onto a special membrane. This can be done using capillary action, where liquid flows upward and carries the DNA with it, or by applying a vacuum to speed the transfer. Once on the membrane, the DNA becomes immobilized, locked in place, so it can be probed and analyzed in later steps with accuracy and stability. Hybridization. In this step, a labeled probe is introduced to the DNA on the membrane. The probe is designed with a sequence that is complementary to the target DNA. Through the natural rules of base pairing, the probe seeks out and binds specifically to its matching sequence. This precise binding allows scientists to identify and visualize the exact DNA fragment of interest, making hybridization a powerful tool for detecting genetic information. Once hybridization has taken place, the final step is detection. Scientists use specialized methods to reveal where the labeled probe has bound to the DNA. Autoradiography captures signals from radioactive probes, producing a visible image on film. Chemiluminescence relies on chemical reactions that emit light, while fluorescence uses probes that glow under specific wavelengths. Each of these techniques makes the invisible visible, allowing researchers to pinpoint the exact DNA fragments of interest with clarity and precision. Advantages of Southern blotting. Southern blotting remains a powerful tool in molecular biology, offering several unique advantages. Most importantly, it is the only reliable method to diagnose facioscapulohumeral muscular dystrophy, or FSHD. Unlike other techniques, it does not require scientists to know the exact location of a genetic disorder beforehand. This method can detect large fragments of DNA with high specificity, ensuring that the results are both accurate and dependable. By using complementary probes, Southern blotting can identify and confirm the presence of specific DNA sequences, making it a trusted approach for genetic analysis and disease diagnosis. Limitations of Southern blotting. While Southern blotting is a powerful technique, it does come with important limitations. The process is time-consuming and requires large amounts of DNA to work effectively. High quality and quantity of DNA are essential, often demanding phenylchloroform extraction, which can be labor-intensive. The use of radioactive probes poses safety hazards, making handling and disposal more complex. In addition, Southern blotting is not usually suitable for detecting mutations at the single base pair level, and accurate sizing of DNA fragments can be difficult. Finally, troubleshooting problems in the procedure can be challenging, which adds to the complexity of the method. Applications of Southern blotting. Southern blotting has a wide range of important applications in science and medicine. It is used for gene mapping and structural analysis, helping researchers understand the organization of DNA. The technique can detect mutations linked to genetic disorders, making it valuable in clinical diagnostics. Southern blotting also identifies viral integration within host DNA, a key step in studying infections and their impact on genomes. In forensic science, it plays a role in DNA fingerprinting, providing reliable evidence for identity verification. Finally, Southern blotting supports research in evolutionary biology, allowing scientists to compare genetic sequences across species and trace evolutionary relationships. In conclusion, Southern blotting laid the foundation for many molecular biology techniques. While newer methods have emerged, it continues to be relevant in specific contexts and has influenced the development of advanced sequencing technologies. I hope you enjoyed this video.